So the dictionary definition of genomics is on the slide that you're seeing, but we're not really going to use that. Genomics is is the study of genomes. A genome is all of the molecular material inside of your cells that defines how your body works and how our bodies work. There are multiple different things that we can study about genomes. When we talk about the structure, what we often mean is the actual sequence of nucleotides, A's, C's, G's, and T's in the genome. So a genome is a long molecule comprised of DNA nucleotides, and we use the letters A, C, G, and T as an abbreviation for these 4 nucleotides. There's only 4 of them. In the human genome, there are approximately 3 billion of these letters strung together. The structure of the human genome is that it's divided into 23 chromosome pairs. 22 of them are identical to one another. There's two nearly identical copies, one from mom and one from dad. And then you have X and Y. And if you're a man, you have X and Y. If you're a woman, you have X and X. So that's another way to think about the structure. And then within each chromosome, there's a little bit more structure we occasionally talk about. In the middle of every chromosome, there's something called the centromere. And at the ends of the chromosomes, there are these special sequences that cap the chromosomes called telomeres. So that's the structure of a genome. But the function of a genome involves many other things. That's what we're really, when we sequence a genome, what we're really getting at is trying to understand the function. So the function is all the things the genome does. So in that long string of letters, we have encoded everything that your body can do. So we have the instructions for how you make all the organs in your body, for how your body develops from a single embryo into all these complicated tissues. And we also have instructions for how your body does things like respiration and metabolism, and even such complicated things as building a brain. Another aspect of genomics is evolution. Evolution is a very broad topic. When we're talking about evolution of genomes, we talk about how genomes themselves change over time, and we usually mean over evolutionary time, that is over very long time periods. We know from having sequenced the human genome and now having sequenced many humans' individual genomes that all of us are nearly identical to one another. So our genomes from generation to generation barely change at all. And most of those changes are small, random changes which don't have any effect. But we can compare our genomes to genomes of other creatures, such as chimpanzees, which are our closest relatives, diverged from us approximately 6 million years ago. And we can compare our genomes to genomes of much more distant things like fruit flies, nematodes, or even bacteria. And when we do that, we discover that we share a surprising amount of sequence similarity even with things as remote from us as bacteria. And when you think about it, that sort of makes sense, because even though we're obviously very different from bacteria, we do very different things, every living thing on the planet uses DNA as its basic code. Every living thing on the planet has to do certain things in common because of that. For example, every living thing has to copy its DNA in order to make copies of cells. So bacteria use a very similar mechanism to copy DNA as humans. So we find the genes in bacteria are, in those cases, similar to genes in humans. And then finally, when we talk about mapping genomes, we're actually beyond mapping. Sometimes refers to just sequencing itself, capturing the sequence. But once we capture the sequence, the first thing we do after that is try to figure out where the genes are. And we'll talk a lot more about that later in the course. But genes, the word gene has changed a lot over the past 50 to 100 years. Today we think about it, one way to think about it is it's a heritable unit, that is something that you can inherit from your parents. Another way to think about it is a small section of the genome that encodes a protein, which in turn has some function. And that's usually what we're talking about in genomics, is the parts of the genome that encode proteins or that encode little bits of sequence that can turn into functional elements. So here's another definition of genomics that emphasizes not just the study of genomes themselves, but also a little bit more about their application. So why are we interested in genomes at all? There are many applications of genomics today, and the list is growing rapidly as we get better and better at sequencing and as we discover more things that we can do with genomes. But it certainly includes medicine, many applications in pharmacy, and if you're not looking at human genomes, agriculture and other areas of science. So genomics is a relatively new field of biology, but it's in many people's minds, it's part of biology. But let me just emphasize a few of the differences between genomics and more traditional biology and genetics, which have a big overlap. So one way you can distinguish genomics from more traditional biology or genetics is that traditionally when we studied genomes or genes, we studied one at a time. Not that long ago, back in maybe the 1980s, it was a common experiment was to study one gene and to spend months or years studying that one gene, or people would spend their whole careers even studying one gene and writing about that gene and trying to figure out what the function of that gene was in, say, people or in, say, a model organism like mouse. But today, because we can capture the whole genome, we often look at all the genes at once, or at large collections of genes all at once. So that would be a more genomics way to study genes. The technology has been the real driver of this, so that's a very big difference, and that's really what's allowed genomics to grow the way it has the past 20 years. In the past, we could only do targeted, what we would call low throughput experiments, studying one gene at a time in, say, one organism. Today, we can do experiments where we simultaneously measure the activity of thousands of genes all at once using the new genomics technology. So that's a big difference between genomics and earlier ways of doing biology. And then the hard part where things differ between traditional and genomics science. In the past, you still had to be clever. You had to understand biology and genetics well. Experiments were not necessarily easy to do. They might take years. Today, some of the experiments, you can still do the same kinds of experiments. Some of the experiments are easier to do, but we generate so much data that the data itself is overwhelming. So because the technology has gotten very clever, very efficient, and very inexpensive, we're now looking at doing experiments where we measure thousands or tens of thousands of genes all at once in multiple tissues and multiple samples. And we know going into such experiments that there's probably a lot of really interesting results that are going to come out. But when we get the data, we discover that, oh my God, there's so much data here that it's hard to figure out where the interesting results are. So we have to deal with big data problems that we didn't have to deal with before. We have to deal with statistical uncertainty in ways that we didn't have to deal with before, and we have to deal with large-scale computation in ways that were never necessary for earlier types of biological studies.